A warm welcome to all. As you can read, this video is for quick revision of all hemat practicals just a day before your physiology practical exam. Contents of this video includes short description about Newbar's chamber and microscope, procedure about hemoglobin, WBC, RBC, DLC that is differential leukocyte count, then blood group and BTCT. Things to recall in Newbar's chamber are: it has depth of 0.1 mm and it contains total 9 boxes. The corner 4 boxes are lined by single line, which we use for WBC count, and central 1 box, which is lined by 3 lines, is used for RBC count. The corner 4 WBC boxes are subdivided into 16 small boxes, and central 1 RBC box is subdivided into 25 small boxes. Amongst these 25 boxes, 4 corner and 1 central is used for RBC count. The volume of 4 WBC boxes is 0.4 mm cube and of 5 RBC boxes is 0.02 mm cube. These numbers are required during the respective practicals for calculation of final cell count. Now, the things needed to recall in microscope are: lens power used for WBC count should be 10x and position of condenser should be lowest. Power for RBC count should be 40x or 45x, whichever present, and condenser should be in middle position. Power of lens during DLC practical should be 100x and condenser position should be highest. Now we'll see the procedure of hemoglobin. The name of the method is Sahali's hemoglobinometry. You need to take first n by 10 HCl in HB tube up to the mark 10 on percentage scale. Then you need to take a prick and suck the blood up to the mark 20 on HB pipette. Then you need to pour this blood in HB tube by blowing through the HB pipette. Gently mix the content in HB tube and wait for 10 minutes. These 10 minutes are required for the production of acid hematine. After 10 minutes, start adding distilled water drop by drop. Continue doing this till the color of solution in HB tube matches the color of plates of HB apparatus. When the color matches, see the level of fluid and note down the reading. Procedure of the WBC estimation starts with the taking small amount of WBC diluting fluid in watch glass. Then take a bold prick and suck the blood up to the mark 0.5 on WBC pipette. Immediately after this, suck the WBC diluting fluid up to the mark 11 on WBC pipette. Gently mix the content in pipette by rolling it under the palm. Discard the first drop and touch the next drop to the edge of cover slip, which is placed on Newbar's chamber. As soon as the drop touches to the edge of cover slip, it will spread automatically below the cover slip. Now wait for some time for cells to settle down and then focus the Newbar's chamber under power 10x and look for the WBCs. WBCs will appear as black dots under the microscope. Count all the cells in 4 corner boxes and multiply it with 50. You will get the WBC count. The normal WBC count ranges between 4000 to 11,000 per millimeter cube. Now we'll see the steps of WBC count in sequence once again. Now we'll see the procedure of RBC count. Take a small amount of RBC diluting fluid in watch glass, which is colorless. Take a bold prick and suck the blood up to mark zero point 0.5 on RBC pipette. Immediately after this, suck this RBC diluting fluid up to the mark 101 on RBC pipette. Now gently mix the content in pipette by rolling under the palm. Discard the first drop and touch the next drop to the edge of cover slip. The drop will spread automatically under the cover slip by capillary action. Now focus the Newbar chamber under power 40x or 45x and look for the RBCs. RBCs will appear as oval, light pink in color with regular margin under the microscope. Count all the cells in 5 boxes within the central one box and multiply it with 10,000 to get the RBC count. The normal RBC count in males ranges between 5.5 to 6.5 million per millimeter cube, and in female it is 4.5 to 5.5 millions per millimeter cube. Now we'll see the steps of RBC in sequence once again. Next practical is differential leukocyte count. In this, you need to take a glass slide, put a blood drop after prick on this glass slide near one of its end. Now take another glass slide, touch its edge to blood drop. Blood drop will spread over this edge. After this, immediately drag the slide to other side. This will prepare a blood smear. Now let this blood smear to dry. After drying, pour the Leishman stain drops over the smear portion only and count the number of drops that has been poured on the smear. Now wait for 1 minute. After 1 minute, pour the equal number of drops of distilled water over the smear and wait for 8 minutes. After 8 minutes, pause the slide but not with directly water falling on it. Now let the slide get dry and put a oil drop on any portion and focus it under the lens power 100x. The identification point for neutrophil is it has 2 to 5 lobe nucleus and the granules are pink color. The identification point for eosinophil is it has 2 lobe nucleus and red granules. And the identification point for basophil is it has 2 to 3 lobe nucleus with dark blue or black color granules. The identification point for large lymphocyte is it has a round nucleus, the occupying whole the cell with thin cytoplasm. Identification point for small lymphocyte is it also has a round nucleus occupying whole cell with a thin rim of cytoplasm. The identification point for monocyte is it has notched or horseshoe shaped nucleus and the size is largest among the all WBCs. In the procedure of blood group estimation, Take a 2 ml of normal saline in a test tube and take a bold prick. Place this prick fingertip on the top of test tube and mix the blood drop with the saline inside the tube by inverting the tube 2 to 3 times. Now centrifuge this test tube at a speed of 3000 rpm for about 3 minutes. After centrifugation, you will see a clear yellow fluid above. You need to discard all this while keeping the red portion. Now take a tile and name the 4 depressions as A, B, C and D. Pour the 2 drops of blood from test tube in A, B, C and D. Now take a drop of antisera A in depression A, 1 drop of antisera B in depression B, 1 drop of antisera D in depression D and 1 drop of normal saline in depression C. Mix the content by gently shaking the tile. Wait for 8 to 10 minutes and then look for the agglutination. Agglutination will look like a clumps as you can see in the depression B. Differentiate it from the Rolex formation. Rolex formation will get disturbed after shaking, whereas agglutination will not. If you get agglutination in depression A and depression D, blood group is A positive. If you get agglutination in only depression A, blood group is A negative. If you get agglutination in depression B and depression D, blood group is B positive. If you get agglutination in only depression B, blood group is B negative. If you get agglutination in depression A, B and D, blood group is AB positive. If you get agglutination in depression A and depression B, blood group is AB negative. If you get agglutination in depression D only, blood group is O positive. If you get agglutination in depression C, that means it is a false positive result, which means there is something wrong with the antiseras. So you need to repeat the procedure with another fresh antiseras. Before starting with the procedure of bleeding time and clotting time, let's see the difference between bleeding time and clotting time. Bleeding time is the time from start of bleeding after prick till it stops through the wiping by filter paper, whereas clotting time is the time from initiation of clot formation in the capillary tube till the formation of fibrin thread. So the bleeding time involves only vasoconstriction step of hemostasis, whereas clotting time involves the temporary plug formation and definitive clot formation steps of hemostasis. In procedure of bleeding time by Duke's method, first take a deep finger prick, then start the stopwatch. After 30 seconds, absorb the blood drop on filter paper. Repeat the procedure every 30 seconds till bleeding stops without pressing or squeezing the wound. Now encircle all spots and number them in sequence. Count the number of spots and multiply the number with 30. At last, divide the total number by 60 for expressing the result in minutes. Normal bleeding time is 2 to 5 minutes. For clotting time by Wright's method, again take a deep finger prick, start the stopwatch, dip one end of capillary tube in the blood drop, keeping other end at a lower level. This will fill blood into the tube by capillary action. Now wait for 1 minute. After 1 minute, gently break off about 1 centimeter bits of glass tube from one end and look for the fibrin thread between the broken ends. Repeat this procedure every 30 seconds till fibrin threads between the broken ends are visible. Now at last count the number of pieces and multiply them by 30. Divide the total number by 60, in which add 1 minute and express the result in minutes. Normal clotting time is 3 to 8 minutes. Thanks and best luck for your exams.